Christophe Le Tourneau. I'm a medical oncologist working at the Institut Curie in Paris. I am the head of early phase clinical trials there. Precision medicine is a way to use all data from an individual to prevent, diagnose or treat. So in oncology, what we call precision medicine is to use data from, I would say, high throughput technologies either to guide therapy or to prevent cancer or to diagnose cancer. Up to now, we have been used to evaluate the efficacy of specific drugs against certain types of cancer. Where we're moving is not to give up this part of drug development, but we are moving to a new generation of clinical trials where we do not assess the efficacy of a specific drug in a specific clinical indication, but where we develop treatment algorithm to guide therapy using high throughput technologies. Meaning, in these clinical trials, what we assess is the value of the treatment algorithm that has been set up. In the treatment algorithm, there are several features: what technology was used, what we were looking at, what were the thresholds, and what is the molecular alterations and drugs matching. And also, what is the prioritization if there are several molecular alterations. So at the end of such clinical trials, you may want to sell the treatment algorithm. So it's not a drug, but it's a treatment algorithm that you want to demonstrate it improves patients' outcome when you use it. So it's a new way, it's a new kind of clinical trials, not evaluating drugs, but treatment algorithms. So we are only at the beginning of that revolution. To date, there is only one randomized clinical trial that has been published, which is the one performed at the Institut Curie and published in the Lancet Oncology recently. That clinical trial was negative for the primary endpoint, but some subgroup analyses are promising, and we are now launching a Shiva 02 trial, which will be the follow-up on the Shiva 01 based on that subgroup. There are also other similar clinical trials ongoing in the US, obviously, but also in Europe and in France. So the Shiva 01 trial was a randomized trial comparing two different populations of patients. What we are going to do in the Shiva 02 trial first is to refine the treatment algorithm, first to use new drugs, but also what we'll do, and is very innovative, is that each patient will be taken as its own control. So meaning that we'll compare the efficacy of matched targeted therapy as compared to what we do in the clinic in the same patient. So the patient will first receive conventional therapy and then matched targeted therapy, and the primary endpoint will be the ratio of the duration of control of the disease on matched targeted therapy divided by the duration of control of the disease on conventional therapy. So what is interesting in this kind of design is that you do not have problem with the heterogeneity of patients' population in two arms you compare, because in fact the patient will be the same receiving the two treatments. We are here because Shiva is a kind of a project where several teams from the research center at the Institut Curie and from the hospital at the Institut Curie, but also several other centers participate in. And I think we're here because it's a nice example of a collaboration between several teams with different expertise in a single project with the aim to improve patients' outcome. The main goal of being here in Boston today is to meet our colleagues from the US, and I already had contacts with some of them to share data. So we got data in the Shiva trial, but there are data generated here in the US, and the goal is to share data, to augment the power of the data to tell predictive biomarkers of efficacy of targeted therapy.